everyone, and welcome to today's live broadcast, From Heart to Mind, Simplified iPS Cell Reprogramming into Cardiac and Neural Cells under Hypoxic Conditions, presented by Dr. Rick Cohen. We are excited to bring you this educational web seminar presented by LabRoots and sponsored by Eppendorf. Eppendorf is a leading life science company that develops instruments, consumables, and services for liquid, sample, and cell handling in laboratories worldwide. Its product range includes pipettes, centrifuges, mixers, spectrometers, and PCR cyclers, as well as freezers, bioreactors, CO2 incubators, shakers, cell culture consumables, and cell manipulation systems. Eppendorf products are broadly used in academic and industrial research laboratories worldwide. I'm Christina Jewell of LabRoots, and I'll be your moderator for today's event. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge that window, just click on the screen icon located in the lower right. Now, if you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window. Or you can use the Q&A button to let us know you're having a problem. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located at the bottom left-hand corner of your web page and follow the process of obtaining your credits. Also, throughout this presentation, you will notice a few polls that will pop up in the middle of your screen. You will have 30 seconds to respond, and we appreciate your participation. I would now like to introduce our speaker, Dr. Rick Cohen. Dr. Cohen is the director of the Stem Cell Training Course and Research Associate in the Department of Biomedical Engineering at Rutgers University. Over the past 8 years, he has been instrumental in providing cell-based materials and hands-on training to participants from both academia and industry. In the past, he directed the Rutgers Stem Cell Core Facility and has trained the first generation of T32 multi-institutional stem cell trainees. He has collaborated with PepperTech in Rocky Hill, New Jersey, to develop several commercialized stem cell media and related products. He plans to continue his efforts to offer engineered cellular solutions to answer questions about cellular reprogramming and differentiation to needed cell types. One of the ongoing projects in Dr. Cohen's lab examines if dopaminergic neuron and cardiogenic differentiation of iPSCs be optimized using small molecules. The aim of this study is to determine the optimized and scalable method to produce functional dopaminergic neurons for studies in Parkinson's disease. The final focus of this project is to seed the cardiomyocytes or their progenitors on a biocompatible and transplantable scaffold in order to create a cardiac patch capable of restoring function to damaged myocardium. For Dr. Cohen's complete bio, please see labroots.com. Please join me in welcoming Dr. Rick Cohen. I will now turn the presentation over to him. Dr. Cohen?
Thank you for that introduction, and welcome to the presentation this morning. I'm going to start off with poll question number 1. Do you currently reprogram cells? And we'll get underway with the presentation. Today's presentation is separated into 3 sections, the first of which will start right away with the generation of human iPSCs with 1 transfection. Before we get into the full presentation, here's a small introduction about human-induced pluripotent stem cells. They can be generated by introducing certain factors, namely OCT4, SOX2, KLF, and c-MYC. In our protocols, we have changed c-MYC to L-MYC. With our polycistronic vector, which I'll introduce a little bit later, we have some other genes that will help us both select and view the cells that are undergoing this reprogramming. Typically, when these cells get reprogrammed, they can be induced to form both ectoderm, endoderm, and mesoderm. In today's talk, we're going to focus on neural cells, specifically dopaminergic neurons, and cardiac muscle. And lastly, if these cells can be generated in a way without using animal products in a safe way that they have been shown to differentiate and not form tumors, potentially these cells can be used for patient-specific cellular therapy. Some of the factors to optimize involve gene delivery method. Do you want to transfer single, multiple plasmids, use chemical or electroporation methods? What kind of cell type do you want to start off with, either a newly established primary cultures or something that's commercial or even transformed? Things like culture medium containing either serum or are defined that are serum-free are important to consider. And lastly, we have gone from a standard CO2 regulated incubator to low O2, which means the levels of oxygen are regulated by pumping in nitrogen. So generating iPSCs with one transfection, optimizing the reprogramming. Here we're going to introduce a novel plasmid. So this is a polycistronic episomal plasmid together with the latest iteration, which contains a MyoD transactivating domain, and that's called the TAD domain, fused to the 5' end of OCT4. And OCT4 is strung together with the other gene using a self-splicing domain called a 2A domain, originally derived from the foot and mouth disease virus, but is found in other types of virus. We have included blastocidin S-deaminase gene fused together with a monomer of red fluorescent protein to be able to both select and visualize. The last thing to consider, if you're going to use DNA, is that your DNA must be what I call ultra-low endotoxin. So if you have a kit, a commercially available kit that claims to have super endotoxin levels, we take that kit and during an intermediate step use Triton X114 to further reduce endotoxin levels. Without that additional step, we find that there's too much endotoxin and we don't get good reprogramming. And this is a depiction of the episomal plasmid. It contains the OREP and the eBNA motif, which are typical to pretty much all episomal plasmid systems. We've optimized by including a small PGK promoter upstream from the eBNA1, but the main cassette is promoted using a CAG promoter. This is a CMV chick beta actin fusion, which robustly expresses the TAD-OCT4, SOX2, KLF, LMIC, and RFP blast. It's followed by a WPRE domain and a beta globin poly A. Upon transfection into the cell, this is translated to RNA within the cell and then made into 4 separate proteins, where you have the 2A domain in this area. It's thought to cause a ribosome to skip and thus make 5 separate proteins. Poll question number 2. What method of reprogramming do you use in your laboratory? And you'll have a few seconds to answer this question before we go on. Okay, let's proceed. Again, generation of iPSCs with one transfection. We're on to point number 2, cell culture optimization. The low O2 tension using a 4% oxygen has allowed us to use this single episome and have very successful reprogramming. When we used standard oxygen, normoxy, which is around 19 to 20%, we got very little transformation. When generating fibroblasts, we also use human serum, not because the human serum works any better than fetal bovine serum. It's because we want to take our research to a translational type of situation, so that we use all humanized conditions, opposed to standard media that uses 2.5 millimolar glutamine. 3 or 4 times as much is needed for robust fibroblast growth. Another point is pre-treatment with the p53 inhibitor, and more recently we're now including a caspase-3 inhibitor, does 2 things. One, it inhibits p53, and if you look in other kits from commercially available sources, they include a separate plasmid or a combination plasmid to interfere with p53 signaling. So here we rely on a small molecule, and the caspase-3 inhibitor helps with viability post-electroporation. And we only include that for a few days. The other thing we do is we've switched away from using Matrigel, which is an animal-derived product, which works perfectly fine. Nothing's wrong with Matrigel. But we use recombinant vitronectin, also produced in an animal-free or a xeno-free situation. So not only do we grow the fibroblasts on vitronectin, but then when we do the electroporation, they're immediately plated and generated into iPS cells on that substrate. And here's a depiction of our protocol. We electroporate using a neon system from Thermo Fisher, using the 100 microliter tip size. Typically, we use 10 micrograms of DNA with 1 million cells. We've gone on to now use upwards of 5 million cells with the same amount of DNA and see almost equally good transfection. We haven't finished those testing to see if we get equally good generation of iPS cells, but I'm assuming because the viability and the red fluorescence are equally as well, we can scale up the amount of cells. That's day 0. They're electroporated and then plated in the same fibroblast media they've been growing in, together with 5 small molecules. The next day, we change the medium to a knockout serum replacer. This is KOSR supplement, also from Thermo Fisher, using a low osmolarity medium or knockout DMEM type of medium, again with 5 small molecules. We now add back pen strep, which we've omitted in the first case, and go to the selection with 5 to 10 micrograms of mil of blastocidin for up to a week. Usually, between 10 and 14 days, we observe something called a protocolony. And in the following slides, I'll show you what that looks like. Essentially, that's a rearrangement of the standard patterns that fibroblasts grow into something else. It's hard to describe otherwise. The last step is after 21 days, where we start to see these more mature colonies, and this may take even up to 30 days. We manually clean away the non-transformed fibroblasts. We use PBS EDTA passaging method, so no enzymes used aside from when we passage the fibroblasts over here. So we go from enzymes to non-enzymatic method. We also change to an animal-free media, which is human serum albumin containing or non-human serum albumin containing. We generally get better performance using it with albumin, at least for the first part. And they're still plated on vitronectin when we passage them. The last part, as I mentioned before, 5 small molecules. Some of these are commercially available as a kit called Boost or Boost 2 from EMD Millipore. Here's our p53 inhibitor, and because I have a publication in process, I can't name the .1L inhibitor, but there are several, and this is crucial. Without the .1L inhibitor, the efficiency of the episomal reprogramming is quite poor. In fact, sometimes we don't get colonies. So by the addition of this last into the 5 small molecule, we get anywhere between 5 and 25 clones in one well of a 6-well dish when we start out with at least 500,000 cells plated in that format. Poll question number 3. Do you already use a low oxygen tension incubator for routine stem cell cultures? And if yes, what percentage of O2 do you use for reprogramming your cells? I'll give you a few seconds to answer this, and then we'll progress on to the rest of the presentation. So if you're interested in a more inclusive and expressive study, there's another application note that I worked on with Eppendorf, as seen in this slide. And the incubator that I use in my lab is similar to the 170R with the 4-split door. The one that I have is slightly different. It's actually an 8-split. So these doors are split into 8 compartments. That actually helps a lot with the minimization of loss of nitrogen and fluctuations of the levels of oxygen and CO2. So using the Neon system, this is 1 day post electroporation. This is the phase contrast. And as you can see, many cells get the RFP. There are some that don't. And what I refer to as a proto-colony, and this may be a little difficult to see, is depicted in this green box now, also in red, where the pattern of the swirl of a fibroblast are changed into this area that looks like something different is going on. And when you follow these, after about 21 days, they appear to be somewhat normal-looking iPS colonies, and sometimes you get overgrowth. When you take these 21 days and you clean them and passage them using the PBS EDTA method, you do get carryover of the feeder cells, which are the same fibroblasts, so they're all along here, that either did not receive the RFP, or if they did receive the RFP or the polycistronic episomal vector, they didn't reprogram for one reason or another. But clearly this is quite large, very well-defined border. Still, it takes 3 to 4 passages to eliminate the carryover feeder layers. And the way we do that is by using the PBS method again. We allow the cell clumps to settle out, and the single cells seem to float, and by successive washes, we rinse away the single cells and retain the clumps. So it's a very straightforward, very easy method to use. These are the same cells, 1 day post-split, and they already forming well-defined colonies. They look much cleaner. And 4 days later, they're expanding. And then this is changed from the animal-free to something called the animal-free low-protein media, which is the same recipe minus the human serum albumin. So we've gone from serum-containing fibroblast media to Kozer, which is a serum product, and that is animal-containing. You can get a human version of the knockout serum replacer, which we have not yet tried. And then you can go to the low-protein media eventually to continually culture your cells. We do some immunological characterization, which is typical to qualitatively describe what's going on to the cells. And here in the top left is DAPI, just a regular nuclear dye. Lin28 and NANOG are also pluripotency genes, which have been described in the so-called Thompson mix that combines OX4, SOX2, NANOG, and Lin28 for reprogramming. In our studies, we use KLF4 and LMIC. So the presence or appearance of Lin28 and NANOG is not from our plasmid. It is from the reprogramming process. So Lin28 is a cytoplasmic RNA binding protein, and nanog is a nuclear transcription factor. And furthermore, the appearance of TRA160 in the bottom left indicates that these are also iPS cells. So in combination, these are three markers that prove our point. When we use flow cytometry, so this is another immunostating protocol. We use it, these cells are grown on vitronectin. And again, this is the animal-free low-protein media, no HSA, similar media with HSA. And we double stain for SSCA4. And that's like TRA160, it's a cell surface marker. And we co-stain that with Lin28. So these are cells that are dissociated off the plate. And in this case, when we do that type of analysis, we use acutase. You can also use triple. We do not use PDS-EDTA because those leave behind a lot of doublets. So when those cells are co-stained under both conditions, we see around 98%. It's a very high degree of purity for these cultures. Poll question number 4. Which lineages do you try to optimize the differentiation protocol for in your lab? I'll give you a few seconds to answer this question before we proceed with the presentation. All right, thanks. Let's go on to the next slide. Now we're going to proceed to the second part, the differentiation of these iPSCs into dopaminergic neurons. The differentiation into dopaminergic neurons, we've decided to work with another group that has collaborated with us and Peprotech and use a small molecule-based combination, 2 ALK inhibitors, activin-like kinases. This is the TGF-beta pathway, SMAD pathway, so 2 inhibitors there. A GSK3 beta inhibitor, and also the combination with sonic hedgehog signaling. Instead of using sonic hedgehog, which when you buy it in a recombinant form is often not modified properly to be robust in signaling, but small molecules will directly activate the pathway by bypassing and activating smoothend. And this is based on the work of Stenerker et al. This is a group in Germany, and they've pioneered this, and now we're optimizing it and changing it slightly to allow the user to have more flexibility. We are using a media from Peprotech, which is like a DMMF12 combined with a neurobasal. There's an N2B27 mix added, which is animal-free at this point, with also L-ascorbic acid. It's combined with these ALK inhibitors, LDN 193189, and the SB431542 compound, again with the smooth agonist, and the GSK3 beta inhibitor here is CHIR 99021. That compound is going to be featured later when we do the cardiac differentiation as well. You may see other abbreviations, B for BDNF, G for GDNF, C for CDNF, A for activin A, T for TGF-beta 3, and 8 for FGF8. We call this DAN, small molecule-based protocol. And as we go through the timeline, you'll see the different days of differentiation or development. The first one is called FPNE1, which stands for floor plate neuroepithelial cell stage 1. And this will take place within 4 to 5 days. We call that the neurosphere formation. Originally, we were using a specialized dish that's treated with a polymer to prevent the attachment of the cells. However, we've gone on now to use the non-TC treated Eppendorf 6-well dish, which is about the same price as a TC dish, much cheaper than a polymer-coated dish, and we get similar results. So we're pretty happy with that finding. Once we develop these neurospheres, and I'll show you pictures of those the next slide, we plate those, and we call those the adherent expansion phase, or the FP&E2 phase. And that goes on for another 4 days. After which, we get to the FP&E3 intermediate stopping stage. And this means they need to be expanded for at least 4 to 5 passages. We use accutase at first, and then go on to the PBS EDTA method, and I'll explain why. But this floor plate stage can be used to develop not only dopaminergic neurons, but other type of neurons. So it does not specify the midbrain specifically. When we're done with the floor plate, we freeze down 10, 15, 20, even 30 ampules of cells and store them away. We go on to the patterning stage, which removes the 2 inhibitors. So now we're limited to the smooth agonist and CHIR in this stage. We call that the DAN1 and 2. The difference is DAN1, when we replate them, we include a ROCK inhibitor to help them stick and survive. The DAN2 just has these 2 compounds right here, smooth agonist, CHIR. Then we go to something called the DAN3 minus. I'm going to forward this so you see the DAN3 plus. The DAN3 minus is based on the DAN3 plus. However, we remove the dibutyryl cyclic AMP and the DAPT, which is a gamma secretase inhibitor, which eventually inhibits Notch signaling. So in this context, when we add CDNF and FGF8, we discovered a novel phase, which we call DAN3 minus. So we're able to robustly expand and amplify the number of incoming cells that have dopaminergic quality. We see expression of TH early without having to go to the DAN3 terminal differentiation. And here's some pictures of what the cells should look like when you have done it correctly. So this is the FP&E1 stage, and what you would expect are neurospheres, but really what happens under this context, we see a sausage or elongation type situation, and if we manually select limited numbers, we plate these out. More or less, we do 2 things. Selection, small scale in a 24-well dish, and then bulk in 10-centimeter dishes. When you do it right, you see patches that almost resemble iPS patches, but they're different, morphologically different. So now we've progressed to the FP&E3 stage and cleaned out some of the cells that are forming more differentiated cells. When we have a culture of FP&E3 where we don't feed them quite as much as we should, we still get the patches, and it may be hard to see, but there are cells in these areas. There's very few DAPI-positive cells, which indicate cell nuclei, but the floor plate stain was engrailed and FOXA2, 2 canonical markers for floor plate. So we're able to make really good floor plate with small molecules. Then when you go on to amplify and then switch them into the the DOPA3 or the DAN3 situation, rather, again, here's nuclei, and this is 500 micron. The scale bar here is 500 microns. We see expression of synapsin that's co-localizing with tyrosine hydroxylase. And this is marker of mature neurons. We also see early on the appearance of PITX3, which is a definitive marker in combination with tyrosine hydroxylase and TUJ1 for immature or committed but not fully differentiated dopaminergic neurons. So this method is quite good at generating the cell. In the literature, there's a PA6 feeder layer based amount of studies where they take this mouse cranial mesenchymal stem cell and use it as a feeder layer for neural cells, and you get robust dopaminergic differentiation. Why that happens, they don't know. So several studies, they use proteomics results and distill down half a dozen to a dozen different growth factors as a more defined method and why the PA6 feeder layer works. We tried to use only those growth factors, but we found we had to use a few small molecules, the typical neuralizing ones, to at least start the cascade for this growth factor-based timeline. Similar to what you saw with the FP&E, this is EC23, a stable retinoic acid analog. So you can use retinoic acid, but be aware it's both thermosensitive and light sensitive. H for HGF, hepatocyte growth factor, SDF1 alpha, VEGF-D for VD. S1 is soluble frizzled receptor 1, frizzled receptor protein 1. It binds Wnt and neutralizes them. P for pleiotrophins, and I2 for IGF2. In this context, we continue to use the PeproGrow neural stem cell media. And again, very similar to the other pathway, where we have a stage called DOPA1, where we generate these neurospheres. We go on to an adherent expansion patterning. And what happens in this stage, some of the neurospheres, when we plate them out, are certainly not of neural differentiation quality. And then sometimes we have to do a little bit of manual cleaning, sometimes more passaging methods. And lastly, when you go to the DOPA3 stage, it's similar as a DAN3 stage, where we include BDNF, GDNF, dibutyrocyclic AMP, and DAPT. We do now include activin A and TGF-beta, as we do find it does hasten the differentiation. But what's interesting, the DOPA2 stage, when we include CDNF and FGF8, we get these robust, very, very small, sub-10 micron cells that respond only to CDNF. And we think those are a novel dopaminergic precursor. So let's go to see what these cells look like. Opposed to the sausage-like cells that you see with the DAN, the DOPA1, these are all circular. So this is retinoic acid and sonic hedgehog. And they should be hollow and small. If you let them get too big, they'll start to get very dark and necrotic in the middle. So do not let that happen. When you plate these out, the spheres stick and rapidly spread out. And lastly, slightly higher magnification, you can see that these cells appear to be very bipolar and long ramified processes. These are typical neural-looking cells. We don't know if they're glial or neuronal, and that's where the recipe, the DOPA2 recipe, helps us expand. And this is immunological characterization of those cultures. So you can see robust synapsin and co-localization with tyrosine hydroxylase. And lastly, I had mentioned the use of FGF8 and CDNF to amplify this progenitor pool. And this has become very important because FGF8 is known to affect dopaminergic cell growth, but there's less known about CDNF. And no one knows why, or no one knows how CDNF works. So we're using this system to kind of characterize the effect of CDNF on the cell. And here's an example of what happened. So when you plate out the cells from the DOPA2 stage, and you let them get very dense, so this is a mat of cells. There's and we've calculated somewhere around 3 million cells in one well of a 6-well dish. These are not whole, these are small colonies. And by the addition of CDNF alone it looks like there's less cells, but all of these patches are highly three-dimensional, and we get somewhere between 70% and 100% more cells in one well. So we can get around 5 to 6 million cells as opposed to the untreated. So clearly, CDNF has an effect, but we don't know exactly what it's doing. So are these cells functional? Great, we've grown these cells up. We can amplify them 2 different ways, but do they actually do something useful? So I'm going to show you data called HPLC, and we're going to look at dopamine secretion. And what I'm not going to show you today, which is ongoing, is a collaboration with Dr. Prabha Smogi and a postdoc, Nicola Francis, where she's going to take both the DOPA3 and the DAN3 cells, so from both methods, and transplant them into animal models of Parkinson's disease using a special matrix. If you're not familiar with HPLC, the data is kind of old-timey looking. It's a trace that comes out that looks like little peaks. And this is a standard mix. So the lab that has the HPLC will put together 25 picomole of norepinephrine, dopamine, DOPAC, which is monoamine oxidase broken down from dopamine. Okay, so DOPAC leads, or it's from dopamine. And then finally, homovinylic acid is derived from that. 5-HT is serotonin, leads to 5-HIAA. And if you're interested in the names, it's 5-hydroxyindoleacetic acid. So those are run, and then a sample is injected. And if it's dopamine, it lines up with this peak. If it's norepinephrine, it will show up here. And so it's based on where the peaks and how high the peaks are. So when we do the studies of neurotransmitter release using cells derived from the DOPA protocol or the DAN protocol, and this is when we change the media, we do a washout. Just by adding Hanks balanced salt solution, we're creating a stimulus, and they're secreting dopamine. And then when you add potassium chloride to get a depolarization, we get further release. What we're trying to do now is figure out a way where we could change into a base media with no other compounds that would interfere with our HPLC without eliciting a change in secretion of dopamine. So both are seen here. And then more recently, we've seen a secretion of serotonin in cells that were prepared using the DAN method, not as much seen with the DOPA method. We don't know what the difference is, but at least now we can tell that maybe we have something else aside from dopaminergic cells in our cultures. The last part of the talk is going to introduce the differentiation into cardiomyocytes. And this is separated into 3 ideas here. Small molecule based, and this is a very simple, straightforward, plate the cells out, change the media a few different times, and if you do it right, you start getting cardiomyocytes within 10 days. We've tried to optimize the use of human serum albumin. We've determined we can use some amount of human serum and or a defined mix of B27. While this seems to be consistent, and we call this the 14-day protocol because nothing really changes, you just have to wait. We were interested to know if we can migrate to some fantasy type of situation where we could make cardiovascular progenitors and then use those for scalability purposes and use those and seed those onto these biocompatible matrices. So right now, I'm going to show you some data using this simple cardiac differentiation, and we call this the 14-day protocol. The base media here is RPMI, 500 microgram per ml of HSA. You can use the same type of dilution or a 1 to 200 dilution of human serum and get similar results, and with quite high amount of ascorbic acid. You take your stem cells, your iPS cells, and use Accutase, plate them with ROCK inhibitor on vitronectin and wait about a week. And they form almost a monolayer. You switch it into the RPMI media containing a high dose of the CHIR GSK3 beta inhibitor for 2 days. Very high dose for 2 days. You remove it, and that goes back just to the base media for 2 days, and then 2 more additional days with a Wnt inhibitor. It's actually a porcupine inhibitor, an enzyme that modifies Wnt and allows for secretion. And we use here Wnt 59C, but IWP2 or 1 or 4, those work as well. And then by day 8, we've seen beating of cellular clusters. So if you go back here to the phase contrast, you can see that there's these clusters, and some of them are smaller, some of them are larger, and it's kind of stochastic. We can't really program the shape of the clusters. These stain for GATA4, a nuclear transcription factor, alpha smooth muscle actin, which is in both cardiac and smooth muscle, and a cardiac specific stain for cardiotin. So together with these 3 antibodies, we're sure we're making cardiomyocytes. We don't yet know if they're ventricular in origin or atrial, but we'll characterize that a little bit later on. Every once in a while, we see in culture these clusters, and this one happened to resemble what we call the beating bunny. And I'm going to play a movie, and you'll see the movie takes about a minute, and you'll see the clusters beat. And that's a representation of what we see with this protocol. Okay, and we're back. So you had an example of what happens with this clusters. And if you were paying attention, you would see a lot of the clusters in this rabbit over here, here, all over were beating. Some of the other areas around here were beating as well. So if we can get these cells on a biocompatible, physically able to move matrix and create something consistent, then we could create this cardiac patch. So that's why in the next protocol, where we call this thing a 2-step cardiac differentiation protocol, is to make these cardiovascular progenitor cells where we can optimize the production of both cardiomyocytes and the much needed vasculature that would help the cells integrate into a real heart tissue. So very much like the 14-day protocol, we accutase and plate the cells, but we don't let them expand in stem cell media. Rather, we go directly from stem cell media into DMEM B27 with insulin. I did forget to mention up front for the 14-day protocol, until the cells are beating, no insulin is added. there are two genes that are important to keep suppressed. And if you add insulin, you get those genes expressed and you get no cardiomyocytes. But since we're making cardiovascular cells here, we add insulin, thioglycerol, a lower amount of LAA, also CHIR, so we're activating Wnt pathways, and also BMP4. We call this mesodermal induction. So the cells from this can be taken to other pathways, not only cardiomyocyte progenitors, and then after these cells get dense, we passage them for at least 3 to 4 times into a similar media, but now include 2 ALK inhibitors, dorsomorphin and AAV301. And we use beta-mercaptoethanol as an antioxidant, and these form a monolayer. And in theory, these are forming these cardiovascular progenitor cells, which the second phase would be taking those and adding the same Wnt inhibitor along with BMP4 and induced cardiomyocyte. So we're at this stage, and hopefully by next year we'll have some good results with the cardiomyocyte progenitors and this pathway. So I can't answer that yet, but hopefully, like I said, in a few months we'll get some answers. So poll question number 5. If you use low O2, what are the benefits you find with your cultures? And you'll have a few seconds to answer this before we finish up. So in summary, we're able to generate iPSCs with one single transfection. Now we do use electroporation. It is very simple. If you have a chemical transfection, you can try that. It's a simplified protocol. It does require small molecules. I've shown you we're able to take those cells and differentiate them 2 different ways. They're related, but they're definitely 2 different ways to make dopaminergic cells. We're in the process of characterizing the action potentials and very much more excitingly from my perspective, what is CDNF doing to these cells? So we're doing some immunostaining to see if they're increasing the proliferation rate, the differentiation rate, and if we get more dopaminergic cells or just more neurons in general. And what will happen to these cells in vivo? And I'm excited to collaborate with Dr. Mogay's group and follow the transplantation of the cells from these both pathways into NOD-SCID mice after they receive a 6-hydroxydopamine lesion. So can they repair Parkinsonian symptoms? And lastly, differentiation into cardiomyocytes. The first 14-day protocol is simple and straightforward, somewhat stochastic. We can't predict what are the shape of those clumps. That's why we're going to the second type of protocol, and determine if the cells we're getting are atrial or ventricular. Apparently, if you include low dose of retinoic acid during the initial stages, very much like the DOPA protocol, you get more atrial origins. Once we get those cells, the collaboration with Dr. Cohen's group in the Rutgers Biomaterial will allow us to create a patch. At first, we'll put them in a 24-well dish and then scale up to a 6-well dish and see if we can create a beating patch. And some of his biomaterials are durable, and some will degrade over time, 1 month, 6 months, 1 year. And lastly, another collaboration with Dr. Bassoon's lab in biomedical engineering is using a fusion of elastin-like protein with different growth factors that form nanoparticles that will hasten the vascularization of these transplantable biomaterials. I'd like to thank you for your attention, and these are some of the collaborators and the people that are supporting and the institutions that are supporting the studies: Peppertech, and I have been working together for several years to develop media, specifically animal-free or xeno-free. The Stennecker lab was supported by Michael J. Fox, and these are studies that were pioneered by Dr. Peter Reinhardt, and he's no longer there. He's at this institute. And Nicole Francis, and she was supported by an NIH grant. She performed the HPLC to transplantation and is probably going to do the action potential. And these are some undergraduates, Asta, and she's part of the Women in STEM program at Rutgers, and Neil, he's also a BME student, and they were responsible for doing the cell cultures, both the neuron and the cardiomyocyte. I'd like you to thank you for your attention, and if you do want to know more about the incubators, there's a whole landing page, and there's not only these larger size incubators, but there are some smaller size versions that they're not quite boutique, but they are, I guess, a 48 liter instead of 170 liter. Thank you again, and I'm going to turn over the presentation to our moderator. And if you have any questions, please push those out as soon as you can.
Thank you, Dr. Cohen, for that informative presentation. As you mentioned, it is time for Q&A, and I want to remind our audience, if you have a question you'd like to ask Dr. Cohen, please do so now. Just click on the green Q&A button at the lower left of the presentation window. Type your question into the box that appears on your screen and click a send button. We will answer as many of your questions as we have time for. Okay, let's get started. Dr. Cohen, our first question is, why do you think hypoxia is important for reprogramming?
Why has to do with certain pathways that we've not completely characterized, that are either not turned off or turned on in lower oxygen tension.
Is differentiation carried out under hypoxic conditions?
Yes, all the studies that you've seen today, we use low O2 concentration at 4%. We don't know if that's optimal for neuron and cardiomyocyte, but we started off with that, and we're going to investigate other levels in the near future.
Can we adapt our protocols to use low oxygen tension?
Absolutely. Just go ahead and lower it immediately. You can try an intermediate or just go right to 4%.
How do I remove the episomal vector subsequently from my cells again?
So the episomal vector is very inefficient and doesn't persist. So if you look at the red fluorescence, you'll see it fade over the course of 21 to 30 days. It's well known that it loses 5% or more efficiency every time the cell divides. So while there's no real proof in this study that the DNA doesn't somehow recombine into the cells, we no longer see red fluorescence. And thus, as other people have said, the episomal vector is inefficient and gets lost after time.
Dr. Cohen, for the two-step cardiac differentiation protocol, at which step do you add insulin in your medium?
So the first step, making the mesodermal cells, you would have insulin. And then when you amplify and grow them as the CVPCs, or the cardiovascular progenitor cells, you still have it. And only when you go to the Wnt signaling inhibition step do you remove it.
Dr. Cohen, we have an... Oh, go ahead.
I have to predicate that. There's very few studies to show that these CVPCs really exist. Go ahead.
Okay. We have an audience member who would like to inquire about what method do you use for freezing cells?
So I published a protocol paper in conjunction with Biocision, and we detail that whole method. And it's very simple. We passage with PBS EDTA, and we don't even centrifuge the cells. So we remove the PBS-EDTA when the cells round up. We gently squirt the cells off and put them in a tube. And for example, if we're doing that to one well and we want to freeze that as one ampule, we volume reduce it just by aspirating the media after the clumps of cells have settled. So we volume reduce it to a 0.5 ml. We add a 2x freeze media, which we make in our own lab, but you can use any commercially available 2x formulation. And then we just freeze it like that. So if people are interested, I think in my bio, I'm not sure if my publications are listed, but if they look up my name, it's Cohen, R.I., I think it's a 2015 publication about freezing cells.
We have some great questions coming in. Here's one. How much time do I have to plan for the whole experimental procedure from iPSC generation up to mature functional cells, for example, into cardiomyocytes?
So typically, the time window to make passable iPS cells using an episomal vector is around 30 days. And that doesn't always include the generation of primary fibroblasts. So for example, if you bought an ampule of fibroblasts from a vendor, we've used Lifeline Cell Technologies. They have a media system that's really robust. Their products are great. We buy that and get it in. We can start doing the transfections within a week, and then with all the time points needed, I said within 21 days you could start to see pretty early cells, and then colonies form, and then really you need 3 to 4 passages. So typically that's another 30 days or another 4 weeks to amplify your cells. So that's 2 months, and then the cardiomyocyte, the 14-day protocol, is just another 2 weeks. So probably 3 months from start to finish.
Dr. Cohen, do you freeze cardiovascular progenitors?
I would like to say yes, but as I said in the presentation, we're able to make mesodermal cells. We're not able to create a stable cardiovascular progenitor cell cultures just yet. So we could passage them 2 times, then they start to die en masse. And so we know we're missing something.
Do you think hypoxia is important at every stage of differentiation?
No, and that's why when you get to the cardiomyocytes, we may have to modify the levels. I think for neurons, I think we can keep it at low. I'm not sure if we need as low as 4%. We may want to go up to 6%, but I wouldn't go any higher than that.
Dr. Cohen, does the CHIR 99021 concentration differ between different iPS cell lines?
Yes, and they differ between embryonic stem cell lines too. So generally, when we pioneer a protocol, we'll take one cell line and we'll use several different concentrations of CHIR. And what we see with high concentration, and that's at 6 micromolar when we're making cardiomyocytes, is we see more clumps. When we use low, and that's about 2 micromolar, we see more sheets. So rather than clumps, the cells form like a muscular sheet. And then we decide how we want to proceed with that cell line. It doesn't mean that those concentrations will work exactly the same for every cell line.
We have had so many great questions coming in, but we are running out of time. This is going to have to be our last question. Dr. Cohen, how to generate iPSCs from LCL lymphoblastoid cell lines?
So you can use episomal vectors for that. I know there are specific protocols to activate blood-related cells prior to the transfection. I'm not quite familiar. There is someone at Rutgers that does that. They happen to use activated T cells, and then they use Sendai virus. But you can use an episomal vector.
Dr. Cohen, thank you so much for this great Q&A session. and your very informative presentation. Do you have any final comments for our audience?
If you have my contact information, and if you have more questions, you're welcome to send me those via email. The plasmid that we're working on, we're making commercially available through a related company, and we'd be happy to share that with people who are interested in trying it.
Thank you once again, Dr. Rick Cohen. And a big thank you to our sponsor, Eppendorf, for making today's broadcast possible. Now, before we go, I want to let everyone know that today's broadcast will be available for on-demand viewing through May 17th, 2017. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now, and we thank you for joining us. Hope to see you again soon. Goodbye.